Ever feel like your life is just go, 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 constantly rushing around trying to be everywhere at once?
Yeah, tell me about it.
Well, get ready to feel a little less stressed. Because today we're diving into a world where rush hour makes our commutes look like a walk in the park.
I'm intrigued. What could be possibly be more hectic than my inbox after a vacation?
Today we're talking about protein transport inside a cell.
Ah, cellular logistics. So we're talking trillions of proteins zipping around at lightning speed, each one with a very important job to do.
Exactly. We've got stacks of research here on how this intricate dance of molecules actually works. And get this, if those proteins don't get where they're needed on time, well, the entire cell could go haywire.
It's like a well-choreographed ballet, except if one dancer is out of place, the whole performance falls apart.
A very high-stakes ballet. Our sources make it clear that cells have these amazingly precise systems to make sure each protein reaches its correct destination. We're talking pinpoint accuracy on a microscopic scale.
It's mind-boggling, really, when you think about it.
Absolutely. So, to break it down, our sources mention different destinations within the cell, like the nucleus, the peroxisomes, the mitochondria. Almost like we're about to embark on a whirlwind tour of a microscopic metropolis.
I like that analogy. And every city needs a bustling downtown area. So let's kick things off in the cell's version of Times Square, the cytoplasm. It's a hive of activity with proteins being made left and right.
Right. But of course, not all proteins are made equal. Some have very specific jobs that require them to be elsewhere in the cell.
Exactly. It's not like they can just hail a cab.
Speaking of hailing a cab, our sources mention the nucleus, the cell's command center. It's like the city hall of our microscopic metropolis. How do proteins with nucleus-bound jobs actually get there?
Okay, so imagine this. A protein needs to get to the nucleus, but it's out in the cytoplasm, like being stuck in a traffic jam, miles from your destination.
In there. So how does it escape the gridlock and reach its destination?
Well, it's all thanks to something called the nuclear localization signal, or NLS for short. Think of it like an exclusive address label, a specific sequence of amino acids that act as a VIP pass to the nucleus.
So no NLS, no entry.
Pretty much. Our sources emphasize just how important this signal is. Without it, the protein's essentially lost in the cellular shuffle, like a tourist with the wrong address.
But with the NLS, it's smooth sailing.
Precisely. The protein is essentially flagging down its own personal taxi service. That's where important comes in.
Important. That sounds important.
It is. Important is the key that unlocks the door to the nucleus. It's a transport protein that recognizes and binds to that NLS address label on our protein.
So it's like important is the taxi driver, recognizing the destination and picking up the protein passenger.
A perfect analogy. But even with a taxi, you've still got to get past security, right?
Right.
Especially for somewhere as important as the nucleus.
Good point. And I'm guessing it's not as easy as flashing a driver's license.
Not quite. The nucleus has its own security checkpoint, the nuclear pore complex, or NPC for short. This is one exclusive club.
Exclusive is an understatement. Our sources describe the NPC as this massive, incredibly selective gateway embedded within the nuclear membrane. It's the ultimate bouncer, only letting molecules with the proper credentials pass through.
Right. And remember that important protein we were talking about? Well, it's holding the golden ticket.
The NLS.
Exactly. That NLS tag allows important to bind to the NPC and effectively guide its protein passenger through this otherwise impenetrable barrier.
So important's got its passenger, it's got the VIP pass, and it's through the door. Mission accomplished, right?
You'd think so, but hold on. The cell's got another trick up its sleeve because remember, efficiency is key here. We can't just leave importance stuck inside the nucleus.
Good point. It's like leaving your taxi driver stranded after they drop you off. What happens to important? How does it get back out to pick up its next passenger?
Okay, so picture this. It's closing time at the nucleus, and importance ready to clock out. But instead of hailing another cab, it gets met by a very important molecule called RAN-GTPase.
RAN-GTPase. I'm guessing this isn't just some random molecule hanging out in the nucleus.
Definitely not. Our sources describe RAN-GTPase as a bit of a molecular switch, kind of like a traffic light that controls the flow of important.
Okay, I'm intrigued. How does it work?
So inside the nucleus, RAN-GTPase interacts with important. But here's the catch, Daft. This interaction changes important's shape, causing it to release its protein passenger.
So RAN-GTPase is like that friend who helps you politely escape an awkward conversation at a party.
Exactly. It's like, okay, important, your job here is done, time to mingle.
Love it. So important's free from its passenger. Then what? How does it get out of the nucleus?
Well, RAN-GTPase doesn't just ditch important after the breakup. They stick together, and actually, RAN-GTPase escorts important back through the NPC to the cytoplasm.
Talk about a true friend.
It's all about teamwork. And once important is back in the cytoplasm, it's free to pick up its next nucleus-bound cargo and start the cycle all over again.
It's a beautifully orchestrated system, really. But I'm thinking about all those other destinations we mentioned earlier, like those cellular recycling centers, the peroxisomes.
Yes, the peroxisomes. Those guys are the ultimate multitaskers, essential for breaking down fatty acids. Think of them as the cell's energy snack bars. And they also detoxify all sorts of harmful substances.
They sound like they're working overtime. So how do proteins know to go to a peroxisome? Another address system, maybe.
You bet. But instead of just one address label, like the NLS, our sources point out that peroxisomes have two main entry points. And get this, each one has its own specific address code.
Okay, wait, 2 doors, 2 codes. It's getting complicated.
It's all about efficiency. These different entry points likely help the peroxisome handle the huge variety of proteins it needs to do its job.
Right, so it's like having different loading docks for different types of deliveries.
Exactly. So the first address code, PTS1, is like writing your address at the very end of a letter at the C terminus of the protein.
Okay, makes sense. And the second code.
The second code, PTS2, is like writing your address at the beginning, at the end terminus.
Got it. So depending on where the address is on the protein, it goes to a specific peroxisome entrance. But I'm guessing important isn't the designated driver here.
Good catch. Peroxisomes have their own set of drivers, receptor proteins called PX5 and PEX7. PX5 recognizes the PTS1 code, and PX7 recognizes PTS2.
So it's like having specialized delivery trucks for different parts of the city.
Exactly. And just like the important NLS duo needed the NPC to actually get into the nucleus, these receptor proteins guide their protein passengers to a special docking station on the peroxisome membrane.
The docking station. Is it anything like the NPC?
You know, that's a great question. Our sources point out a key difference here. The peroxisome uses something called a docking ring complex for import. It's much simpler and smaller than the massive NPC we saw earlier, but it gets the job done.
It really highlights how much variation exists in these transport systems, even within a single cell.
It's incredible.
And speaking of incredible systems, we haven't even touched on the mitochondria yet, the powerhouses of the cell.
Right. They're often called the powerhouses, but our sources emphasize that mitochondria do so much more than just produce energy.
Really? Like what?
They're crucial for controlling reactive oxygen species, which, if you didn't know, are unstable molecules that can damage the cell.
Oh, wow. They're like the cleanup crew. Anything else?
Yeah, and here's something that might surprise you. They even played a role in programmed cell death.
Wait, what? Programmed cell death? That sounds a little ominous.
It's actually a good thing. It's essential for maintaining a healthy balance in the body. Think of it like removing old, worn-out parts so they don't cause problems.
Makes sense. But now I'm really curious, how do all these proteins involved in such vital processes actually get into the mitochondria? Doesn't it have, 2 membranes to get through?
You're right, it does. And that brings us to another fascinating twist in our cellular adventure. To get into the mitochondria, proteins actually have to be unfolded.
Unfolded? Hold on. Doesn't a protein need its specific shape to actually function?
It does. That's why they get help folding back into the right shape once they're safely inside. But think about it, to get through a narrow doorway, sometimes you need to temporarily fold up a large piece of furniture, right?
Ah, okay, I see where you're going with this. So the protein needs to temporarily fold itself to squeeze through the mitochondrial membrane.
Exactly, and the mitochondria has a whole team of helpers to make this happen.
Of course, there's a team. There's a team for everything in the cell. Who's on the mitochondria's moving crew?
Okay, so first up, we've got chaperone proteins, specifically a type called HSP70.
HSP70, those sound familiar.
They might. Remember how we talked about chaperones helping proteins fold correctly? Well, HSP70 plays a similar role, but this time it's making sure the protein is unfolded correctly for its grand entrance into the mitochondria.
So it's all about teamwork, even at the molecular level.
Absolutely. And HSP70 doesn't work alone. It teams up with another key player, the mitochondrial targeting sequence, or MTS for short. Think of the MTS as the protein's backstage pass to the mitochondria.
So like the NLS for the nucleus.
Exactly. It's a specific sequence of amino acids that acts like an address tag, signaling signaling, hey, this protein belongs in the mitochondria.
Right. No MPS, no entry.
You got it. So HSP70 recognizes the MTS on our protein, and it's like, all right, buddy, time to get you ready for your big debut. Let's get you unfolded.
It's like a pre-show ritual.
Exactly. And once the protein is temporarily unfolded, ASP70 escorts it to the mitochondrial membrane. And here's where things get really interesting.
More interesting. I didn't think it was possible.
They encounter a remarkable piece of cellular machinery, the TOM complex.
Another complex. What does this one do?
Think of it like the mitochondria's very own VIP entrance.
So, like the nuclear pore complex, but for the mitochondria.
Exactly. The TOM complex is the gatekeeper, recognizing the MTS address tag on the unfolded protein.
Okay, so the protein, even though it's all unfolded and kind of vulnerable, is still able to show its ticket to get into the mitochondria.
Exactly. And once the TOM complex has checked the ID, it helps thread the protein through a special channel in the outer membrane and into the intermembrane space, which is like a waiting area before the main event.
Okay, so the protein is through the first checkpoint, but now it's in the intermembrane space. What happens next? Does it just refold on its own and casually stroll into the matrix? So the protein is through the first checkpoint, but now it's in the intermembrane space. What happens next? Does it just refold on its own and casually stroll into the matrix?
Not quite. Remember, it's still got to get through that second mitochondrial membrane to reach its final destination.
Right, the matrix. It's like going through airport security, then realizing you have to go through customs too.
Exactly. And just like at the airport, there are helpers along the way. Once our unfolded protein reaches the inner membrane, it encounters another protein complex, this time called the TIM complex.
Okay, so first there's Tom, and now there's T. It's a lot to keep track of.
I know, right? But the cell's got it all figured out. The TIM complex is like the final gatekeeper, making sure only proteins with the proper credentials get into the matrix.
And I'm guessing that MTS tag, our protein's backstage pass, is still essential here.
Absolutely. The TIM complex recognizes the MTS, and here's the really cool part. It works in sync with those HSP70 chaperones we talked about earlier.
Ah, the chaperones are back. They're like those super helpful flight attendants who guide you to your connecting flight.
A perfect analogy. So as the protein is threaded through the TIM complex into the matrix, the HSP70 chaperones are there, ready to lend a hand, or should I say, a molecule.
So they're helping the protein refold correctly once it's inside the matrix.
Exactly. Remember how we talked about the protein being temporarily unfolded to squeeze through those membranes? Well, now that it's reached its destination, it's time to put it back together, like unpacking a suitcase after a long trip.
It's like those HST70 chaperones are the ultimate organizers. They make sure everything is in its proper place. But what about that MTS tag? Is it still needed once the protein has been refolded?
Another great question, and the answer is nope. Once the protein is inside the matrix and folded correctly, the MTS is like that boarding pass you don't need after you've reached your gate. It gets clipped off.
Gone. Recycled, maybe?
Yeah.
The cell is so efficient. It's truly remarkable how everything works together so seamlessly.
I know, it's incredible, and we've only scratched the surface of this intricate world.
I believe it. But even with just this glimpse into the cell's transport system, it's clear that protein trafficking is absolutely essential to life as we know it. Every heartbeat, every breath, every thought. It all depends on these tiny molecular machines being in the right place at the right time.
Well said. It makes you appreciate the complexity of even the simplest things in biology.
Absolutely. So, next time you're stuck in traffic, just remember it's nothing compared to the hustle and bustle happening inside your cells 24/7. And on that note, we'll wrap up this deep dive into the fascinating world of protein transport. Thanks for joining us.